In the previous video clip, we reviewed the structure of DNA, the main unit of genes. In this video clip, I'm going to tell you about how to create genes. There are 4 complementary structures that control genetic information. But where did those 4 structures in the genes come from? So living organisms can create DNA components on their own to some extent, but with poor efficiency. So living organisms, including ourselves, solve this problem with one solution. We eat other organisms. To pass down the genes, living organisms eat other living organisms, indigest the DNA components, break them down, and rearrange them into their own genetic codes. So otherwise, living organisms cannot pass down their genes. So let me give 2 examples. The one is inosinate, what we call 5'-IMP. So this is the umami ingredient of dried bonito. The dried bonito is used in Japan as a soup stock. And the other one is guanylate, or 5'-GMP. This is also an umami ingredient of dried shiitake mushroom. A dried shiitake mushroom is used as a soup stock in Japanese cuisine, or some other cuisine as well. So guanylate that represents the genetic letter G. So this is a component of the DNA already. So these 2 acids are sometimes listed as a nucleic acid-based umami materials on the food labels. So in general, the mixtures of inosinate and guanylate are used as seasonings. The inosinate is converted into guanylate or adenylate inside our bodies and constitute part of our DNA. So that's why inosinate is so tasty. So then, look at this one. So inosinate can be converted into adenylate or guanylate. Then adenylate or guanylate are components of DNA. So that's why we love inosinate here. So let's look again at the structures of DNA-based pairs that control genetic information. I told you about these in the previous video clip. Then GC-based pairs have 3 hydrogen bond interactions, and AT-based pairs have 2 hydrogen bond interactions. And AT and GC pairs are properly formed based on these hydrogen bonding interactions. However, AT and GC pairs are not necessarily required if we consider the complementary compounds that form the pairs through hydrogen bond interactions. You can design some other pairs, or you can come up with some other ideas. Why do we use AT and GC? The answer to this question is simple. Because these pairs form spontaneously on the primordial Earth. In the 1950s, Dr. Miller was a graduate student at the University of Chicago in the United States. Then the intention of his doctoral research was to prove that the materials that served as the origin of life can be synthesized from the simple materials in the environment of the prebiotic Earth. To stimulate, so these are the condition that, so these are the material that he started with. To stimulate the condition on the primitive Earth, Dr. Miller put water, ammonia into a flask filled with hydrogen, and methane gases. They boiled the flask and exposed it to the electrode discharges that imitated the lightning. Then also put UV there that imitated the sunlight. And surprisingly, after a few days later, the liquid became brown. The careful analysis showed that the two different amino acids were formed in the liquid. Then after continued experimentation, at least 5 different amino acids had formed, and also a little bit of the purines. Then at that time, this experiment drew much public attention because it suggested that the proteins may have been created spontaneously in the primitive Earth. Now, let's consider the origin of the genes. In 1960s, Dr. Ogel and others mixed water with hydrogen cyanide and ammonia and heated the sample, the simple heating here. Then he got adenine with pretty high yield. So today, adenine is considered to be the first DNA base formed on the Earth. That's why adenine is a component of NAD+, FAD, ATP, coenzyme A, and other compounds that we have in our body. You can see the adenine structure very often in the structures of the compound that we have in our body. The other components of DNA may have also been created spontaneously. Let's take a look at this. So how about the sugar part of DNA? The Professor Fisher did experiments a long time ago. Then he put aldehyde and water. Then he looked at the reaction. In the presence of calcium, he got the formation of fructose. That's a sugar. So then he got a mixture of D and L fructose, but this is shown here. So the sugar can be described like this. So then if you get this kind of reaction, you get furanose, 5-member ring. Then, but if you get this reaction, you get pyranose, that's 6-member ring. You can go either way. Then actually, usually you get more pyranose than furanose. So yield is 70% here, yield here is 30%. Okay? So this one is easier to make. However, we use 5-member ring for the DNA structure. Why is it? People actually synthesized both to see what happens. This is the furanose DNA, the natural DNA. Then this is pyranose DNA, 6-member linked DNA, right? So then people compared these two, they found that with this structure, DNA base is facing this way. But in this 5-member ring structure, DNA base is facing this way. With this conformation, you can see the base pairing. But with this, base is faced in a different way. That's why you cannot see the DNA base pairing. That's why we don't use 6-member ring sugar for DNA structure. We use 5-member ring. So as these components gathered together and interacted in a complementary way, genes were probably formed. And eventually, living organisms came into being. That is how scientists think at this moment.